I'm just starting the recording.
Okay.
Yeah, sure.
With your permission, Dr.
Yeah, go ahead.
Krishnamurthy, Dr. Ghosh, and others, let me start the proceedings by introducing Dr. Kannan Krishnamurthy. He's a distinguished biotechnologist. He was the Vice Chancellor of the Nagaland Central University. He was also the founder, Dean, and Professor of Biotechnology, Director of Students' Welfare at the Indraprastha University. He introduced biotechnology as an elective subject for CBSE that made a great transformation in the country. Coming to his association with industry, he was director in SPAN Group of Industries, Associate Director of Biotechnology, Ranbaxy Laboratories, and quite a few other things. But let me flag for your attention that he is internationally acknowledged for establishing the first evidence for the extracellular role for ubiquitin in hematopoiesis. Hematopoiesis means blood cell formation from stem cells in the mouse. This was the first record in the world, and it explained the mechanism of bone marrow transplantation. This remains as a landmark work that he has done, and it has had its implications on a number of research projects taken up subsequently. I would, in passing, mention he has been given numerous awards, and he has had collaboration with universities abroad. In short, we have a very distinguished person who is going to talk on a subject which may not be touted quite often, but then it has a lot of significance because the person about whom he's going to talk is a person whose contributions have had far-reaching implications. And Dr. Kannan was saying that we need to highlight the work of some of these stalwarts who are out of sight, out of mind, and forgotten. But their work continues to have an impact on the ongoing research in the country. So with these few words, let me welcome Professor Kannan for his webinar on the topic for the day. Professor Kannan.
Yeah. Thank you so much, Dr. George. We are very fortunate to have Dr. M.R. Rao, whose mother was the sister of Yellapragada SubbaRow's wife. So welcome, M.R. Rao. And we have Dr. P.K. Ghosh, who has been one of the pioneers of building the Department of Biotechnology. And we have so many other distinguished, and I see Jagannatham is there. A whole lot of people are here. Welcome, and I hope, I'll start the session by sharing my slides, and then we'll begin. Just 1 minute. I thought today happens to be his birthday, Dr. Yellapragada SubbaRow. He was born on 12th January 1895, and he passed away in 1948. Many of us, because when you look at his name, unfortunately, he put it as SubbaRow. Because the editor of the journal where he published his first paper combined SubbaRow as one word and made it SubbaRow, and that made him look like somebody from, Latvian country. So for a long time, many people didn't know that this was an Indian. Until Sri S.P.K. Gupta, whose photo is there, he passed away on 29 January 2023. Who had just finished his journalism degree, and he was looking for something, a biography to do on scientists, and he bumped on the death news of Yellapragada SubbaRow when he was posted as a foreign correspondent in Moscow. And so he traced the whole thing. He did a lot of work, spent his own money, traveled around, and then wrote this book. And this is called In Quest of Panacea. And today I'll be just covering whatever is done is mostly from the work which SPK Gupta did. He wrote several books, and recently the latest book he has really got released is a much better version. I can give you, whoever is interested, from where to buy. It's very enjoyable. To cut a long story short, why is very important? Imagine that, just to give you a brief background, that biochemistry just started evolving only around, say, biochemistry's evolution only started around 1890s, when it was shown that that if you break open the cell wall of yeast and you can convert from the extract sugar into alcohol, what is known as fermentation. That began studying the contents of the cell. But more important than that was that if you look at the average human age before 1900, it was 25 years. But now we know that the age has gone to 70 years. So what is it that has to go from 25 years to 70 years? So what are the drugs, vaccination, how they have played an important role is very important. So if you remember, biochemistry was always considered as part of chemistry. And for a long time, we just had classification as organic and inorganic, and later physical chemistry. That's what led to colligative properties, etc. And the standard was if it's a molecule, it has to be crystallized. And everybody did carbon, hydrogen, nitrogen, and arrived at this thing. And most popular was finding out the melting point and boiling point. This was the story in the 1900s. Then in 1950s, we saw that biochemistry was behaving different than the typical chemistry because they were dependent upon pH and temperature. So this is a very important one, and you will see that Can you kindly mute, because there is some voice coming. May I request somebody whose voice is coming to mute, please? Yeah. So if you look at pH, for example, when penicillin was made, it was quite unstable. But when it was extracted and then put it at pH 6.5 buffer, suddenly it got stabilized for years. So that is the significance of pH as far as the many stable molecules are concerned, and the rest is history about penicillin. So that's why pH, temperature, these play a very important role. And when it comes to protein, temperature, all of you know, if the temperature goes up, the protein gets denatured. Now, if you look at biochemistry in those days, it was happening in very few places. For example, in Caltech, we had Linus Pauling. In Harvard Medical School, there was Otto Folin, whose famous method of Folin-Share-Calto for measurement of protein. Then we had a very big group in Rockefeller University who did cell biology, cell fractionation, protein structure, etc. Then if you come over to England, you have University of Cambridge, where Lawrence Bragg did the X-ray crystallography, and others in Cambridge University looking at metabolism of macromolecules. And then when you come to Sweden, you see that they were developing techniques. For example, say, the analytical ultracentrifuge was developed by Theodor Swedberg, and Arne Teselius developed the Teselius electrophoresis. Then the German biochemist Otto Warburg developed a lot of techniques to measure, oxygen uptake and things like that. And in France, Louis Pasteur was developing a microbiological world and various applications. So this was the scenario in the 1900s, to 1950. And SubbaRow lands up in Harvard in 1921, and so he had So there are very few people in the field, and he was in Harvard Medical School, which we'll be reading about. So if you start looking at it, SubbaRow essentially wanted to become a sannyasi in his early life. But Ramakrishna Mutt did not allow a child to become sannyasi until the mother gave the approval. His mother refused to give approval. So the next best thing, Ramakrishna Mission advised him, why don't you do medicine? So he went on to do medicine at Madras Medical College. Now, that was also the period in the before 1915 and all that period. We found that there are a lot of freedom movement going on, and he was a great fan of Gandhi. So apparently the story goes that he went with a khadi gown for the surgical exam, and the British examiner failed him because you are wearing a khadi gown. So he never got his MBBS degree, but he got only licentiate practice. Then what happened? In between, he survived by a disease those days known as Sprue, and he miraculously escaped due to Ayurveda. So he got interested in Ayurveda, and there was a Ayurveda medical college, in just 1 minute. Okay, so you will see that, he joined this thing, Ayurveda Medical College, and he's learned a lot of Ayurveda, and he was the first man who was trying to, make some relationship between modern medicine and Ayurveda. Then he found that it was not sufficient, and he decided to go to Boston to study at the Harvard to study the tropical medicine. So he went to do a diploma degree there. And then he decided to do a course in biochemistry, and and he started working from 1921 in Harvard. And he was given a problem to work on phosphate estimation. And the phosphate estimation, just one. And phosphate estimation, he was very excited about. And the problem with phosphate estimation before he started was, Folin directed him to work with a man called Cyrus Fiske. And they were trying to develop a method how to estimate phosphate in blood and urine samples. So the test they had developed took more than half an hour to develop color, because color chemistry was the most popular. So you will see that he found a compound, a naphthalene sulfonic acid, which suddenly could give color in a few seconds. So that was a major breakthrough, and he published this paper in JBC in 1924. And later I'll talk about it, because one of the second most cited paper in the biochemistry field, estimation of phosphate and its ramifications. Later, during his PhD, he worked on muscle contraction. He discovered creatine phosphate, and creatine phosphate discovery, isolation, character, that is what earned him PhD. When he was working on muscle contraction, he also discovered ATP. So in 1929, he discovered ATP, and for all this, he got a PhD from the biochemistry department in 1930. Now, 1930, but unfortunately, he never got a faculty position in spite of having phosphate estimation paper, discovery of creatine phosphate, which disproved that Nobel Prize given to Meyerhoff and A.V. Hill was wrong. And ideally, it is actually creatine phosphate which is involved in muscle contraction. So, but unfortunately, those days, citizenship in USA was not given. In fact, I'm told the first citizenship for foreigners came only in about 1949, and he was no more there before that. He passed away before that. So you have to appreciate that he, in spite of doing brilliant work, he could not get a regular faculty position. He got some teaching research assistantship, which was, which is given for fresh graduate student. Anyway, he continued doing that. So he became a teaching fellow in the Harvard Medical School. Then everybody started talking that all this work has happened due to the brilliant student Yellapragada SubbaRow, and his boss Fiske had nothing to do. So everybody used to say it is all the work of SubbaRow, and that created some rift between Yellapragada SubbaRow and Fiske. And in 1935, when Fiske was scheduled to become professor and head of the department, SubbaRow said, I renounce all the credit for the phosphocreatine. It should all go to Fiske. Anyway, that didn't help very much in the whole story. I don't want to get into that. And in 1940, by the time he was helping Lederle fractionate liver. And Lederle had just got into this business of doing pharmaceutical sciences, because this is basically a part of American Cyanamide Corporation. We started a division, and they found that they have been clandestinely taking the help of SubbaRow for various fractionations. So in 1940, he left Harvard Medical School and joined the Lederle laboratories. This Lederle laboratory now is under Pfizer. Okay. So, but I'm talking 1940s. And first thing he attempted was to make penicillin, because there is a very big demand for penicillin during the World War II. So apparently, from what I heard, that the British scientist who made penicillin, when he visited his lab, he said, you have made a better quality penicillin than what we have made. But however, due to patent thing, SubbaRow refused to take any credit for that penicillin work. Then he started looking. Then he said he started looking for one, trying to find out the liver extracts, and started working with that. And he discovered folic acid from that, and he found that folic acid was responsible for the Sprue disease in India. And he discovered that, so he could solve that question, which many people died in India. He said it is due to folic acid deficiency. Then he also, in 1946, he tried to find a broad spectrum antibiotic Aureomycin, which he succeeded. And, out of that came Tetracycline, the second generation, etc. Also, he also worked on, elephantiasis, because he had a training in parasitology. So he discovered this Hetrazan, and you will be surprised, even today, that is the first line of treatment available for elephantiasis. Then, when Harvard Medical School, the cancer department, AML children were suffering from. So Sidney Farber, who was heading the lab, he was good friend of Yellapragada SubbaRow. So he took folic acid and gave children who were suffering from acute myelogenous leukemia. Suddenly, the disease spread much faster than possible. So SubbaRow said, okay, I'll make an antifolate. So he made the antifolate, which is the Methotrexate, which all of us know. And then suddenly the cancer could be cured. So in the history, he was the man who first showed that cancer is treatable. Unfortunately, he didn't live long. So in 1948, he passed away. The reasons is debatable. So as you can see, the contribution which I was telling, so he has worked in analytical methods, which is what is your, phosphate estimation, etc., creatine phosphate. Then natural product synthesis, because he synthesized folic acid. Then he worked on bacteriology, virology, parasitology. Okay. And then he worked on cell culture. He did clinical trials also, because he was in Harvard, and we'll discuss about that. Then cancer. And then, that is, so in such a short span of time, he contributed to so many fields. So the most famous is the colorimetric estimation, as I said, by Fiske and SubbaRow. And, many of us had chance to use that technique. And mind you, it has survived till the method was modified somewhere around 80s. So pretty long innings for a technique to survive for so long. So we'll discuss about that more. Okay. Then the next one he made was folic acid from the liver extract. So as you can see, SubbaRow in 1945, vitamin folic acid initially saved people from tropical sprue and microcytic anemia. 50 years later, at the turn of the century, expectant mothers are advised to take the vitamin regularly before planning pregnancy to stave off the neural tube defect such as spina bifida. Latest medical reports suggest that folic acid reduces homocysteine levels, a risk factor for coronary artery disease. So, a discovery, this folic acid is a very interesting one because he not only isolated it, he characterized it, he synthesized it. So he did the whole thing. I think in one issue of JBC had 12 papers. So that is the level of productivity he showed at that time. Then another thing he isolated was vitamin B12. Of course, ATP is a history and creatine phosphate. Though ATP is a very unfortunate story in the sense that there was an international conference in Harvard, and at last moment, he was asked to present the work. So it never registered in the book. So what happened was the German scientists who were there and who saw that he was talking about creatine, they went back and did it and won the Nobel Prize. So this is one of the unfortunate incidents which happened. And similarly, creatine phosphate, that was his PhD work. And I think for some odd reason, Cyrus Fiske was very disturbed by all these things, that probably he became mentally unstable. That's what people say. Then when you come to parasitology, so elephantiasis is such a common one in India, especially, wherever the people work in the rivers to wash their clothes and all that, they get mosquito bite, and that is through which the filariasis spreads. So he discovered this compound called diethyl carbamazine. So diethyl, pioneered by SubbaRow, took on filariasis, the scoring that leads to elephantiasis. After years of hesitation, because of unpleasantness with high doses, WHO found in 1998 that a single dose keeps blood free of filarial worm for a year and has deployed DEC worldwide to mass filaria eradication. So it's quite a credit for him that after so many years after his death, it has become a WHO drug. Then coming to cancer, the methotrexate, which everybody talks about, and many people don't know that SubbaRow was the brain behind that. So, as you can see, aminopterin, a 1948 brainchild of SubbaRow, has the folic molecule with an amino radical replacing the hydroxy in the vitamin. It reverses the vitamin action of folic acid. It extends life and improved quality of life of children with acute leukemia and initiated chemotherapy of wide range of cancers. Methotrexate, the modified aminopterin, improved survival of young women suffering from choriocarcinoma, the cancer of the afterbirth mimicking pregnancy. 50 years later, it is standard in treatment of rheumatoid arthritis and psoriasis. It has been successfully used also in treating ectopic pregnancy, chronic ulcerative disease, and can reduce steroid depending of asthmatics. So that is his final molecule which he played around. Now coming to phosphate estimation, as you can see, I told you the naphthol sulfonic acid was the one which made the success, and luckily the dye industry was using this, so this was available in good amount, 1 to 4 commercially available. So he used that and developed a very rapid test to this thing. So estimation time was less than 5 minutes, and reliability up to 1 part per million he could achieve at that time. So as you can see, it became so, because once you know phosphate, you can say whether it's a DNA, RNA, a whole lot of things are possible. All phosphate compounds can be identified by this. It is interesting to see that when creatine phosphate was discovered And when he tried to do phosphate estimation of that by earlier workers, they found that phosphate level keeps on increasing in a frozen muscle. So many people said probably creatine phosphate interferes or is not useful for estimation. Phosphate estimation method may not work for creatine phosphate. And when he analyzed it completely, he found that no, what is happening was whenever muscle contraction happens, creatine phosphate gets hydrolyzed to creatine and phosphate, and so the phosphate keeps on increasing. But once it is relaxed, it gets consumed. So it is a cyclic reaction which is happening all the time. So, and he also discovered that ATP has 3 phosphates and there is a labile phosphate. So, and he developed the method to estimate it in 30 minutes. So the nature of the inorganic phosphate in muscle or both are involved. And he published this work in Science in 1927. This was a PhD work. So as you can see, a man being in Harvard between 1925 to 1948, he had done so much of work that I don't think we can talk of any other scientist who would have worked on so many areas in one's life. And mind you, Lederle, we are talking about 1950s, the turnover was $277 million. Okay, and then slowly it went up to 322 before Pfizer took over. So Lederle made a major role in this thing, and that is even now in the New York, his bust in front of the Lederle lab, which is the Pfizer now. So people have commented on him. So some of the comments are, some of the nucleotides isolated by SubbaRow had to be rediscovered years later by other workers because Fiske, apparently out of jealousy, did not let SubbaRow's contribution see the light of the day. George Hitchings is one of SubbaRow's former colleagues. Life of Dr. Yellapragada SubbaRow is an example of how religion and tradition need not necessarily be incongruous and incompatible with scientific temper, also how they can foster modernity and development. Dr. SubbaRow is an anachronism in today's world, which is more intensely individualistic rather than the world he lived and accepted. This is Dr. Vaidyanathan again. So I'll stop over here, and we can. The floor is open for discussion. Yeah, George, I leave it to you.
Dr. Kannan, can we see? See you a bit more clearly.
Huh?
You had gone. Your face was not fully seen. Okay, now the presentation is open for discussion. Before that, would anybody like to make special comments, Dr. Ghosh or Dr. Redana?
Yeah, Dr. George, let me first congratulate KK Biotech for
and SBPI and SBPI
and BPI for coming forward with introducing one of the renowned scientists that India has produced, Yellapragada SubbaRow. And let me also thank Dr. Kannan. In fact, he has gone through the entire history of Dr. SubbaRow's contributions and really gave a very good presentation on his contributions. I think many of the young scientists should, unfortunately, not many are here, but I think so. University of Hyderabad introduced the Yellapragada SubbaRow Memorial Lecture, which is being given every year on his birthday, and for many years, I think it has. So then, the Federation of Asian Biotech Associations were a non-profit organization. I'm sure many of some of you may be knowing, but for others, it is a non-profit organization that we established almost 20 years back, to provide a platform for the academy, industry, and government to come together and promote innovation and entrepreneurship, those kind of things. And over the years, we have seen that uh, skill development is very important because the graduates coming out of the colleges and universities, they do not have the right skills. So for that, we launched the FABA Academy and started offering the industry relevant skills among the students. And third one was the FABA entrepreneurship, where we are trying to promote a startup ecosystem. These three are them. And the most important thing is to recognize the contributions of the young scientists. FABA has launched Yellapragada SubbaRow Memorial Awards. One for young scientists below the age of 35 with 5 lakhs cash prize. And then the best doctoral thesis for innovative work that they have carried were for two scientists, one boy and one girl, with 1 lakh cash prize, which we have been giving for the past 2 or 3 years. So FABA has, in fact, is trying to recognize and then promote such kind of innovations. And I think Yellapragada SubbaRow we thought was the right person and with whose name we should give these kind of awards. And let me thank Dr. George. Just a few minutes back, he has sent me the link, and I'm really fortunate to hear Dr. Kannan and meet all of you also.
Thank you. Thank you, Professor Reddy. Now invite Dr. Ghosh. Dr. Ghosh.
I am seeing Dr. Ghosh also.
Unmute, Ghosh.
Unmute. Please unmute. Yes, Dr. Ghosh.
Yeah, unmute. After that, you request Dr. M.R. Rao to speak a few words. Yeah.
Yeah.
Yes.
Hello, everybody.
Hear you, sir.
Yeah. Actually, when we read the history book of science, we find that the ATP was attributed as the ATP was discovered by the German scientist first, and later on from the America in the same year, a paper was published on the same subject, and there the name of SubbaRow appears. So when the Nobel Prize was given, it was given to the scientists of Germany as well as the scientists of America on ATP. The point I'm trying to make is that when we talk about ATP structure determination or ATP's quantity estimation, SubbaRow certainly did a great work. But then when we are talking of the history of science, we must state very clearly who did first and why it was done. There is no doubt in my mind that Dr. SubbaRow was an extraordinary scientist. But we have talked about his contribution in the discovery of several molecules that Dr. Kannan had mentioned. Even diethyl carbamazine citrate has been mentioned, which is now an accepted drug for microfilaria infection cure. It does not cure the infection of adult filarial microbe, but it kills it kills the microbes when they are in the microfilarial stage.
Hello.
So the point I'm making is when we discuss science, we must discuss science with a very open mind, without any prejudice. If I give glory to somebody, certainly we should, but we should not belittle the fact that the science is developed by many people, and every contender has to be mentioned in the proper perspective. Thank you.
Yeah. No, in fact, when you read the book, it has been very clearly mentioned that when the ATP discovery happened, there was a conference in Harvard where last moment they were asked to present the work. And then World War broke out. So at that time, his paper was not mentioned in the abstract book. That became a controversial point, and the German scientists were present there at that time. So they were also trying to characterize that. And when they said this is ATP, so they went back quickly, did it up, and published the work by the time these people got around to doing it. So it is a very unfortunate incident. But both of them are equally creditable. They were very close to it. Probably one helped the other without one acknowledging the other. That's only. So it's okay. That's history. M.R. Rao, could I request you to say a few, because he happens to be SubbaRow's, Yellapragada SubbaRow's wife's sister's son. Okay,
Very good.
I had to get somebody from that.
This is SubbaRow's sister's son.
Now, please,
You know, actually the thanks are going to SPK Gupta, who did a lot of research and then brought out the life of SubbaRow to the world. And his book, Inquest of Panacea, is a monument. This is the basic standard book. It's available on Amazon.
Correct.
And because a lot of misinformation is there on SubbaRow's work and his life. Then another thing is another small. This is a big one with a lot of scientific information. Another for common man. This is a book, Life and Letters of Dr. SubbaRow, because he wrote a lot of letters. Even he dissolved his work in department to the president of Harvard University, saying that I'm only a helping hand to the Fiske, and credit goes to Fiske. Though the entire work was done by him, but he gave credit to the Fiske.
Correct.
Oh, I am not a biochemist. I am a statistician and a management consultant.
Yeah. No, but it is very interesting that SubbaRow found that the vitamin B12 gets absorbed onto the charcoal. And the way he developed method to elute it out is very interesting.
Yes, correct.
It helped many others to this thing. So he did a lot of good organic chemistry work in spite of being a medical doctor by profession. So it's amazing that he worked on so many areas.
Harvard has helped him a lot because he went there to do research on medicine, but he became biochemist in Harvard.
Yes.
The opportunities came there.
Correct. So,
and his letters were interesting to see written to his father-in-law about his details work into life. And people think that he became a Christian.
It isn't true.
He wrote a letter to the bishop, to the father, saying that I can't change my religion. I believe in my Hinduism.
Correct.
Those letters are there in the book.
Yeah.
Available in Amazon.
So he was well versed in Sanskrit, German, also
fact,
Quran,
as I mentioned,
Bible, all that he has well read because that's what is reflecting. And Jews were very fond of him.
And
the Jewish
Jews are very fond of him. In fact, Jews have written a very good arbitrary on him. And actually, in Madras, he went to Ramakrishna Mission, did his comparative religion. He did Quran, he did Bible, he did Bhagavad Gita. Comparative religion in Ramakrishna Mission, Chennai at that time. And then he learned astrology also there. Astrology also. Astrology. He predicted about his son's death also at that time.
Correct.
Before he went to America, his wife was pregnant. And he writes from there, don't bother, you will not survive.
Because it died. The child died due to one of the bacterial infection.
Yeah. And he died within 9 months in the womb and 9 months in the world. He predicted it earlier.
Yeah.
So he's a versatile man, and then his views, in fact, his philosophy and then views are expressed in his letters.
Yes.
And I suggest him, if you can get this book,
Sure.
Mrs. Gupta has got this book, you can give it to them. And if anybody wants in Delhi, they can go to Gupta and then get it. If you're in Hyderabad, I can send me your mail address. I'll send the book.
In fact, Dr. S.P.K. Gupta's contribution is amazing. He went around.
But he brought the SubbaRow's life into the world.
Yes.
Otherwise, people don't know him. And a lot of misinformation is there before that.
Yes. No, he went and sat in Lederle and did the archiving work.
What happened was he wrote some 1 or 2 articles. Then Lederle Laboratories Bombay office called him and then gave him a scholar 6 months vacation. He took vacation and went to America, interviewed all the people, Harvard University professors, Lederle Laboratory employees, his colleagues. 6 months he was in America to do research.
Yes.
And all the information is provided in this book.
Yes.
And in fact,
it is a fact actually that emperors and empires have always tried to diminish the work done by the natives. India is always considered to be native. So this kind of attitude, as you are mentioning, Dr. Kannan, which happened to this great man, is likely. Because I find that wherever the empires and emperors have actually come forward, they have always tried to project their work as the highest work.
I know he mentions
this happened to Hargobind Khurana. Hargobind Khurana is one man who has been troubled the most because I have known people who have worked with him, the Britishers, and they have told me there is no scientist like Khurana who has the tenacity to survive the bickerings of the white man,
Amen.
And when he got the, when he worked on the code and came out, the Americans could never digest. Wisconsin University, a lot of them were very upset.
I know.
Because Arthur Kornberg brought him to Wisconsin those days to do nucleotide synthesis.
That's right.
So it's Khurana is another great man.
Great man. Great Indian man.
And still win a Nobel Prize. And go to MIT as the highest paid professor at that time.
That's right.
He said, I'll come to MIT if I'm the highest paid. No one else will get more salary than I do. So these are all...
SubbaRow feels he never put his own credit because in all the papers, his name is alphabetical at the end.
Yes.
And he says research cannot be done by one person. It is a teamwork.
The teamwork. Correct.
That is philosophy.
Especially his work on Aureomycin is very interesting, how he led the team.
In fact, the man who is an agricultural scientist, he brought him to Lederle and asked him to verify all these things.
Correct.
He brought youngsters and then not knowing the subject, trained them and then got the research done.
No, there are some who worked with him and got to become president of Workshop and Dome.
This is the book which gives a lot of information.
Yes. So is there any more questions? Otherwise, we'll wind up today.
I'll have just one comment. Can I make?
Yeah. Jagannathan, please do.
Actually, Yellapragada SubbaRow in his birth centenary during 1995, when P.V. Narasimha Rao was the Prime Minister of the country, a postal stamp was released in his commemoration of his greatness and his work.
100 years. 100 years of his. Right.
100 years of his this work.
Yes.
So Gupta forward it is.
And they brought out the stamp also.
Yes, postal stamp was brought out at that time.
Postal stamp was brought out. Only thing, people tried very hard to get him a Bharat Ratna. Somehow it didn't work out.
Yes.
No, he deserves it. He deserves it actually.
So Dr. Jagannatham is retired scientist from CCMB, and he is the one driving the FABA Academy activities, running this skill development activities after retirement.
In fact, Jagannatham started his career with me in CCMB.
Oh.
I recruited him in 82, 83, if I remember.
Yes, 83.
I'm so glad because I made him go and do MSc in JNTU when he was a junior scientific assistant. Evening, he used to go and attend the MSc class and got his MSc, then his PhD. So, for me, Jagannatham is very special.
Thank you very much, Kannan,
Let
for
me
your
also
support.
introduce Dr. Amit Agarwal. I think Redana, you may not know about him.
Yeah. I was just listening to him before the start of the meeting. So he was mentioning about the myelin sheath.
He just had a Cell paper, 55-page paper in Cell.
Yeah, please forward a copy of that to me. See, in
Let me
GPS
briefly
patients.
tell you interesting history of Amit Agarwal. You know, he joined our BTech biotechnology program in 1999.
Okay.
After 6 weeks, he got selected to Madras Medical College for MBBS. And he decided not to join MBBS and continue biotech.
Just to add that how
it was a very
much
big decision he took.
just to add how much Professor Kannan was.
Can I ask a question to Dr. Amit? Can I?
Yeah.
Yes, please.
Actually, in old age, like our age, the myelin sheath of our nerves always get destroyed, and thereby we develop pain in the different joints. I am actually at the moment suffering from a little dislocation of my hip bone. Now, doctors say that your nerve myelin sheath is spoiled. So my question to the doctor is, can I revive my myelin sheath? They said, we do not know. So I am asking Dr. Amit, is it possible to stimulate the cells so that the myelin sheath is produced and therefore the nerve is again closed and insulated?
A very good question. I think
Amit, Dr. Amit, before you answer, in case of this GB patients, this GB syndrome, no? In that
Guillain-Barré syndrome, you mean?
Yeah.
Yeah.
In that, the people lose the sheath instantaneously, but to get back, it takes several months or years. Yeah.
GB is a very, very interesting disease. It's an example of molecular mimicry. GB, Guillain-Barré syndrome. So what happened was that it comes from when you consume chicken, and that's how it was found. Because some of the chickens which are sick can carry some bacterial infection, which if not very properly cooked transfers to us. And what happens is that the outer cell wall of some of the bacterial protein is very similar like myelin protein. So when the bacterial infection happens, very specific type of salmonella, which is usually found in the chickens, they don't usually come to us, but then if we consume it, it can come into the human system also. And this basically our body attacks, and this memory B cells now after some time start attacking our own myelin sheath. And that becomes the initiation of the autoimmune disease. And that is what Guillain-Barré syndrome is. And you said it very correctly, because since it is of the autoimmune nature, that the attack happens very quickly, right? Since till the B cells are not engaged, it is all fine. But as soon as the nature of our immune system is, as soon as they start the guard, they attack. And that's how they destroy and it's a very disastrous disease. If somebody gets it, you are dead. And to get the myelin sheath very quickly is difficult as you age, just because of the nature of these cells. So the good thing is that myelination is one of the procedure by which our nerve cells can regenerate. So if a neuron dies, it cannot be regenerated, but the myelin forming cells regenerate. The biggest question is that why these cells, even if they are there, they cannot make myelin. And that's what we are working on, and we have some ideas around it. So maybe give or take 2 more years, give it to me, and maybe I'll come with an answer that why the cells are sitting there and they don't make myelin. And that will really transform at least the myelin field. And we have really good ideas around it, basically. So yes, it has been a very big question in the field, is that there, even if you talk about stem cell, it does not work very well, because you can again bring the cells, myelin-making cells. But my question always is that myelin-making cells are already there. By stem cell, even if you bring more myelin-making cells, they are not making myelin. So there has to be something which they are lacking. And that's what we are working on, that what are they missing? And that will allow them to really help us in many, many ways, basically. And as I would say, as Dr. Ghosh, you asked the question related to myelin. I think that myelin is something really so special, is that you can, even by exercise or so, generate more myelin in our brains. So those people who are cognitively very, very active, even in the old age, we keep our myelin alive. So it's good. So all the professors here, you are in a good shape, because by design, we are nerds and we are cognitively very active. Even if we stop moving, we will keep our brain moving.
Amit, I am just making a statement that, I am a vegetarian, so I do not eat chicken. So therefore, the proteins that I am actually a little digressing from today's discussion, but it is a very interesting discussion for many people who are of our age. I am already 83. So therefore, what I am doing is, the doctor who saw me said that you take painkiller. So I am taking painkiller, but he has suggested certain exercise, and you have just mentioned that exercise stimulates, perhaps stimulate the cells, and really I am getting very good result by doing exercise as suggested by the doctor. So thank you, Amit. One day maybe if I am in Delhi, I will try to meet you, because you are an excellent scientist. I found I guessed from the talk that you had given in the beginning. Thank you.
So thank you very much. And just to add it, include a bit of vitamin C, because the cells which make myelin in the peripheral nervous system, which is your issue, what you mentioned, they require vitamin C ascorbate to make better myelin. So, and vitamin C is good. You cannot overdose yourself with vitamin C. We were talking about vitamin B12. It's water soluble, so even if you take extra, it will not harm you.
Okay. Dr. Kanan. Dr. Kanan.
Dr. Amit?
He is in Heidelberg University. So he is waiting for his tenure professorship now.
Oh, okay.
In Heidelberg Medical School.
Yeah. 2 points there. So, Professor Adhanom, I would request you one thing from my side. I don't know whether my student will like it or not. The first author of this paper is Ram Reddy. He comes from Hyderabad.
Yeah, and in fact, the chief operating officer of FABA has graduated from Heidelberg University.
Okay.
Dr. Jagadish Gandla is with us now, running all activities of FABA.
Activities. So perhaps I think you can invite, if he is coming in a couple of weeks, to visit his parents.
Please.
I will ask Ram to write you email. Then he can tell you the story, because he's the guy who was working behind the bench doing these experiments. And also one more thing, Professor Kanan, just to add, is that perhaps in your slide, when you talk about Warburg, you should not use any more Kaiser Wilhelm Institute name, but perhaps you should use Max Planck Institute. And I tell you because this institute is just throw away from our institute. So Otto Warburg worked in Heidelberg, and he's credited for working out glycolysis. And so he's credited for Otto Emden-Meyerhof pathway.
Emden-Meyerhof, all that, yes.
Why I said is that we should remove Kaiser Wilhelm Institute name and this could be really problematic in Germany, especially. I cannot use it also because it is association with Nazis. So when after the Second World War, Kaiser Wilhelm Institute name was changed to Max Planck Institute, and that happened to be the institute where I did my PhD. So I know the history of it. So it will be just Max Planck Institute for Medical Research.
Okay.
But yeah, perfect. So that is really amazing to be really associated also at Heidelberg, to be associated with such kind of histories, that when you just walk around the corridors of the institute and you see that how glycolysis was worked out, and that time it's also enlightening. And I will include, I don't give biochemistry lectures because I'm a physiologist and not a biochemist, but so maybe next time when I'm in a talk, and if some due credits are missing for creatine phosphate and ATP, I can raise hand and say, sorry, we also have to give credit to other people, even if they did not get Nobel Prize. And as rightly said, the due credit has to be given, even if the Nobel Prize has not been given. So that is very important for people.
Yeah, I will get your contact details from Professor Kannan or Dr. George. Yeah. Oh,
perfect.
Thank you very much.
Thank you very much.
So we have reserved a slot for Dr. Amit.
Yes, yes. You can take the date from him now itself. Whenever he is available, I don't know.
So
Work out.
why not Sabba take the credit? You can join as a partner in organizing that webinar.
Then, okay, you go ahead and do it all three.
All three of us,
Okay.
no problem.
Yeah, all three. Why not?
Pharma and
Sure,
TK
most
Biotech.
welcome.
That makes it very strong.
Yeah.
Yeah,
No,
definitely.
we have to slowly form a network to promote things.
What do you
we
say,
can,
Dr. Ghosh?
we can reach out to large
Certainly,
number of
certainly. I
students.
mean, you see, a society actually goes up only when there is crosstalk, when there is collaboration. And
Yes.
if we have more collaboration with a free mind, we will certainly prosper.
Then
Yeah, certainly.
Yeah.
that means we agree that the next one would be hosted by all three. And
Yes.
the coordinates can be worked out by Dr. Redana.
Yes.
Yeah.
Yes.
I'll also
Yeah,
talk to Jagannatham also. He's their coordinator, so I could work out with him.
Okay.
Yeah,
Wonderful.
yeah.
I am actually flabbergasted to listen to Amit, because I liked very much his talk when he started talking about the myelin sheath. Thank you.
Yeah.
I can
Jagdish,
give you more crash
Dr.
course on
Jagdish,
him. It's an
Dr.
amazing thing.
Jagdish is here. Let us hear from him. Jagdish?
Kandla? Is Dr. Bindu here? Bindu Day?
No, she hasn't joined today.
Yeah,
She
Dr.
joined-
Jagdish, are you around?
Joined for a while. I saw her join.
Yes, sir. Yes, sir. I know.
Jagdish is there. Yeah.
Hello, sir.
You
Good
know
evening. I know
Amit?
Dr. Amit.
Dr. John, I'm very much here.
Oh, yeah.
I know
Okay.
Dr. Amit.
Sorry. Bindu,
I
yeah.
joined little late
Sorry.
and I have- It's
Apologies.
okay. It's okay. I have bad cough, so I joined little late, but I was listening to the conversation. I think the conversation is very interesting. And we look forward to listening to Amit. Basically, he's a neurobiologist or neuroscientist, and you remember how we were involved, in building up NBRC. Amit, we look forward to your talk, surely.
Thank you very much. I would.
Jagdish, maybe.
Yeah. Good evening, sir. Hi, Amit. How are you? Hope you remember me. I was with Rohini when Chika and Heinz.
Exactly. Yes.
I was still in
100%.
the interview, if I remember correctly. Yeah.
Yeah, you
So-
are well connected with the Heidelberg neuroscience community.
Yeah, my PhD is with Rohini.
Yes, exactly. You
Yeah.
do the Rohini.
How is Heidelberg doing? How is your work?
Well, it's doing good. We are doing good. We have some really nice story coming up for pain and glial cells. So we will do some non-opioid research. So yes, it's really a very productive time for our laboratory. So many things are popping up now. So good
Are you
timing.
still at IZN?
Yes, I'm part of the IZN. Anyone who is doing neuroscience in Heidelberg will be part of this IZN
Yeah.
umbrella, for sure.
Okay.
And
But you are in which
and,
institute, Amit?
I'm at the Institute of Anatomy and Cell Biology.
Oh, with the Thomas Kuhner. Thomas is also there,
Yes,
right?
exactly. So Thomas Kuhner's lab is second floor. My laboratory is on the third floor.
Oh, cool. Nice to get back to the, my alma mater. It's been 3 years I'm back in India.
Oh, wow. So you've been, okay. I heard about you around that time, but yes, 3 years back in India. Not bad. Where are you based these days?
I work with Professor Redana, so we run something
Ah,
called.
so you are in Hyderabad.
Yes.
Yeah. I think Jagdish, Dr. Ram Reddy apparently is coming to India. Who
Okay,
was,
sir.
The first author of the Cell paper.
Okay.
Maybe let us.
So you should. I would recommend you to grab him. He's shy and he does not want to. He's a diehard experimentalist, I would say. So he's very shy. So you have to grab him and give him a talk, and then he will be very excited to tell what is going on, and then you can poke him technically, right? What things are going on. But a very good hands. So if Hyderabad University grab a very good experimentalist, you should try.
Okay.
Though I don't know how to
I'm
convince.
thankful Ram Reddy has finished.
Yes, Ram is finished, and he got actually the topmost grade. He got the.
He got Samarkand Lord.
Very good,
So
sir.
only 3 to 5% of the entire PhD student in the whole Heidelberg University gets it. Well, Jacob, about this. So yeah, he graduated with summa cum laude, and it was also a very proud moment for myself because he was my first student.
Your first
I
student,
recruited
right?
him. And not only that, but he's also first in his entire family from very much close by that he has got PhD. So it's also a proud moment for him also. Like he comes from a very, very humble family in Hyderabad, around Hyderabad, not exactly. So it's really nice that this guy has really flourished well and come out to be doing very well, good stuff.
That's good. Happy to hear this, good stories after so many years.
Exactly. It takes time. Good science take time.
You as a PI, you know the professional story, right? We as friends, we know the other side of the story, how things
exactly.
go.
Exactly.
Yeah. Good. So, Amit, now what we do at Farber or Whale Tank is that, sir, good evening, sir. Good evening, George, sir. We wrote a book chapter in your book. Happy to
That's one of the authors.
Yeah. So more or less what we wrote in that book chapter is our line of work, sir. We connect industry, academia, government, policy. We upskill a lot of students. So far, past 5 years, my work with Farber, we upskilled around 30,000 people. A lot of them got placed. And then slowly we ventured into venture capital ecosystem. We are helping biotech companies to raise money. And we ourselves launched a VC. So far, we invested on 127 crores. And we have a manufacturing site in Genome Valley. We're kind of building an ecosystem, which like where, any part of the life science ecosystem, it could be startup, student, entrepreneur, they all should be get help. It's all under the blessings of Professor Redana. So he is the connector. He is the deal maker. We are just, executioners in the back. That's it.
We hear a lot of good things about Farber because when I wrote in, George's book, which he edited, we do mention one paragraph on Farber and what work they are doing.
Yes, sir.
So, because all of us have to recognize what others are doing.
Thank you very much.
Thank you, sir. Thank you. Yeah.
So, George, you want to close it now?
Yes. I think it's been a wonderful event, wonderful interaction, and we had the bonus of quite a few unexpected guests. And that makes it all the more interesting. And I would say that this tripartite or the coming together of three organizations today as a sequel to this webinar is certainly most welcome. And I think we will be able to enlarge our footprints and also spread it across. And Amit is a good catch. We will not leave you. We'll keep you under the scanner and look forward to hearing from you. With these few words, let me once again thank Dr. Kannan. In fact, it was he who, popularized this idea that we need to remember those people who are not with us, but who have made outstanding contributions. Now, when you look at the history of Professor SubbaRow, there has certainly been, I would not like to say discrimination, but he has been unlucky. He was not given the due recognition. I was doing a search on AI on SubbaRow. So many interesting things were said about him, like his work led to the work of others, leading to Nobel Prizes. All those things are written in the AI chat GPT, So you get a lot of interesting information. And thanks, Professor Kannan, for laying it out for us. Well, it's a teaser, I would say, to learn more about such scientists. And I'm sure we'll do well in that respect. Thank you once again for joining. And Dr. Kannan has not only been the speaker, he has also hosted the webinar, in the sense, letting in people, asking them to keep quiet. Anyhow, things have been really nice. Once again, thanks to all of you. And Amit, do I say good night or good morning?
Good afternoon.
You can say good afternoon.
Good afternoon to you and good night to all the others. Thank you so much.
Okay. Bye.
Next meeting, Amit, after a long day. Are you still in Dawsonheim?
Yes, still in Dawsonheim.
Okay.
Good place.
We'll stay
Thanks
in
for
touch.
connecting, Jagdish. Then
Sure.
let's be in touch.
Yeah.
Bye-bye.
I'll get your contacts from Ram Reddy. Yeah.
Yes, exactly.
Yeah. Bye.